Unit 3 Free Living Nitrogen Fixing Microbes. Module name Cyanobacteria as Biofertilizers 1. Diversity, Identification and Isolation. Myself, Ms. Triveni Tari from PSS RSN College of Arts and Science. Outline includes diversity and identification of cyanobacteria, isolation of cyanobacteria. Learning outcomes distinguishes cyanobacterial classes, cites examples of cyanobacteria, describes the process of isolation of cyanobacteria. Cyanobacteria, also called as blue-green algae, are photoautotrophic prokaryotic algae. The nucleus is incipient type and they lack membrane-bound organelles. Cell wall is made up of microfibrils. The principal pigments includes chlorophyll A, C-phycocyanin and C-phycoerythrin. Membrane-bound chromatophores are absent and the pigments are found embedded in thylakoids. Many filamentous membrane possesses specialized cells of disputed functions, known as heterocysts, which are usually referred to function for nitrogen fixation. Many species of blue-green algae fix the atmospheric nitrogen in moist soils. Hence, blue-green algae inoculants has been recommended as biofertilizers for flooded rice in many countries. Now, with respect to habitat, we see diversity in cyanobacteria. They can be aquatic, wherein we get freshwater forms as well as marine forms. For freshwater, we have the examples as Oscillatoria and Uvullaria. For marine, we have Trichodesmium and Dermocarpa. For terrestrial, we have the examples as Oscillatoria and Nostoc. They are also found as endophytes, as in the coralloid roots of cycas, then in the thalloid lobes of Anthoceros, and so on. So here we have Nostoc and Anabina, which are found as endophytes. Identification of blue-green algae. This can be done with the use of thallus morphology, wherein we get to see unicellular forms which can be oval or spherical. We also get to see colonial forms, wherein the cells after division remain attached by their cell wall and they are covered by the gelatinous matrix. The colonial forms can be non-filamentous or they can be filamentous. In the non-filamentous forms, we have different morphological colonies which are formed, which can be spherical, cubical, squarish, or they can be irregular. For the filamentous form, they remain in a single row of cells and they will be covered by mucilaginous sheath and they will be referred to as filaments. Common cyanobacteria which are used as biofertilizers include Anabina group, which are heterocystus strains with a thin sheath without branching, do not form mucilaginous colonies of definite shape. The second group is of Nostoc, wherein it is heterocystus strain with thick sheath without branching, forming mucilaginous colonies of definite shape. Third one is Olocyra group, wherein they are heterocystus strains with a thick sheath, usually without branching, and do not form diffuse colonies on agar medium. The fourth group is Citonina group, wherein they are heterocystus strains with false branching without polarity, forming velvet-like patches on agar medium. Now, when cyanobacteria are used as biofertilizers, there are different steps one needs to follow, which includes isolation of cyanobacteria, preparation of starter culture, mass culture of blue-green alga, storage, and the field use of blue-green algal inoculants. Now, we'll see in detail the isolation of blue-green alga. 5 grams of topsoil is taken in 100 ml of Fox medium in a flask and is incubated at room temperature with the illumination of 1500 lux. Further, loopful of this culture is transferred to 10 ml of water in a tube and is shaken well. Serial dilution and inoculation of every dilution is done in Fox medium. After sufficient growth is achieved, microscopic examination is undertaken. If the culture is pure, then further steps will be undertaken. If it is a mixed culture, then serial dilution step is to be repeated. Now here we can see the composition of Fox media, which is the main medium used for the culturing of blue-green algae, wherein the pH is maintained at 7. Starter culture is nothing but once the pure culture is identified, the blue-green alga are grown separately in Fox medium. Enough light is provided to the flask to favor the algal growth. And these algal cultures, which are referred to as starter culture, will be used for initiating mass culturing of blue-green algae. These are some of the online and the offline references you can use for your further reading. Thank you.